Today, let us discuss another important cellular component of blood, which are the platelets. You definitely know they are otherwise called thrombocytes. And what is a thrombus? It's a clot. So, platelets are especially important in blood clotting, and they are crucial to prevent blood loss after vascular injury. So, the number of platelets will be more after an accident or a surgery, and after a fever, the platelet count will decrease because of the development of certain antibodies in response to viral antigens, and hence hemorrhage occurs as a complication of many fevers like dengue fever. So let us move into the topic. Blood has a plasma part and a formed element part. You know that. Formed elements are RBCs, WBCs and platelets. Platelets are the smallest among the formed elements. Normal value of platelets is about 2 lakh to 4 lakh cells per microliters of blood. Next, let us discuss about the formation of platelets. The process is called thrombopoiesis. Platelets, like any other blood cells, are formed from the hematopoietic stem cells of bone marrow. The stem cells are of myeloid stem cell lineage called megakaryocytes. They are giant cells. Platelets are derived from fragmentation of cytoplasm of these giant megakaryocyte cells, and hence they are non-nucleated. They are small cells of about 1 to 4 microns in diameter and have a very short lifespan of about 7 to 10 days. They are colorless and refractile. Next is regarding the shape of platelets. So normally in the circulation, platelets move as inactivated form. See, there are many factors in the circulation as well as in the endothelium to make the platelets inactivated, because activated platelets always enhance the production of clots. Inactivated platelets are hence oval or disc shaped. They have an open canalicular system. You know what is a canalicular system? It is formed of numerous endoplasmic reticulum communicating with the cell membrane, and this canalicular system, this extensive canalicular system will help the platelets to extrude its granules when they get activated. Then activated platelets are spherical. There occurs dilatation of canalicular system. Why? Because in order to extrude the contents of the granules into the exterior of the cell. Then they develop pseudopods because they require pseudopods to move into the particular site. They require pseudopods to move into a particular site of endothelium rupture in order to stop bleeding, in order to stop the flow of blood through there. And then there are other organelles present in the organelles will be present in the center of the cell. Rather, in inactivated platelets, they will be distributed all over the cell. But in activated platelets, due to the development of pseudopodia, the organelles will be present in the center of the cell. Next is about the structure of platelets. We have already studied that platelets arise as fragmented part of cytoplasm from the giant cells called megakaryocytes, and hence platelets are non-nucleated. But these platelets have a cell membrane, then cytoplasm and microtubules. Regarding the cell membrane of the platelets, very important. Cell membrane of the platelets is peculiar and unique. It has again the the bilipid layer. The lipids will be in the form of phospholipids, cholesterol and glycolipids. Here, see, the phospholipid part is important. The phospholipids in the cell membrane of platelets, they are called procoagulant phospholipids because they help in coagulation. So, the platelets are in the inactivated form, they will not expose these procoagulant phospholipids. They will be located in the inside leaf. We have studied that during activation, platelets will change its shape to the spherical. So, at that time, these procoagulant phospholipids get exposed. And then many clotting factors will have affinity to these phospholipids and this will enhance the process of coagulation. So phospholipids are important in the context of coagulation. Then it has carbohydrates in the form of glycocalyx. Then it has proteins in the form of glycoproteins, which is very important. See the structure of the cell membrane there. You can see many glycoproteins acting as receptors for different things like von Willebrand's factor. We will study about it in detail in the coming slides. Then ADP, thrombin, etc. All these things will help in intercellular signaling. That means from one platelet to the other platelet, then from platelet to the endothelium, then from platelet to the subendothelium. All these glycoproteins will help. Then we have already discussed that there is extensive invaginations in the cell membrane and these invaginations will help the platelet to extrude its contents at the time of need. Need is a very important thing. See, platelet is needed means there is a disruption in endothelium. There is a disruption in endothelium means a blood is lost. So, platelet need is a big important thing. Studied about the cell membrane, then comes the microtubules. They forms a ring around the cytoplasm. See the figure. And this microtubules help the platelets to change its shape from an inactive shape to the active form, from a disc shape to the spherical form, and also this microtubules provide adequate support to the cell. Then cytoplasm contains different cell organelles like mitochondria, lysosomes, etc. And that's all about the structure of platelets. This is very important. Chemicals in the cytoplasm of platelets. So, there are contractile proteins, actin and myosin. What is the importance of contractile protein, actin and myosin? They help to change the shape. So, they will help the microtubules to change the shape from inactivated disc shape to the activated spherical shape. Then there is thrombasthenin. It is also a contractile protein and this thrombasthenin is especially important in clot retraction. Clot retraction means squeezing of clot so that the endothelium will come together. The disrupted end of the endothelium will come together and it will aid in healing. Then von Willebrand factor, very important. It is an adhesive protein. We have discussed about adhesion proteins. Von Willebrand factor is an adhesive protein and this protein will help in adhesion of platelets to the subendothelium. So, in order to adhere to the subendothelium, what should happen? The endothelium should be disrupted. So when the endothelium is disrupted, these von Willebrand factor will help the platelet to adhere to the subendothelium. Then fibrin stabilizing factor. The importance of fibrin stabilizing factor we'll study later. It is clot stabilizing factor. Then platelet derived growth factor. That's a very important factor. That factor, that secretion will help in the healing of the ruptured endothelium. Then platelet activating factor. One platelet cannot form a clot. So there should be more platelets. So the platelet activated group of platelets will start attracting the other platelets with the help of this platelet activating factor. Next is a very important thing. There are two types of granules present in platelets. They are the alpha granules and the dense granules. You will have to study all the things present in alpha granules and dense granules. We have discussed everything except serotonin, ADP and calcium. ADP and calcium is especially important in blood coagulation steps, and serotonin acts as a local vasoconstrictor. So when there is a disruption in endothelium, means when there is a rupture in blood vessel, the platelets start secreting serotonin, and this serotonin will act as a vasoconstrictor. So there will be a localized vasoconstriction, and this helps in reduction in blood loss. Properties of the platelets are 3 A's: adhesion, aggregation and agglutination. We will discuss each of these individually in the coming slides. So the first property of platelet is the platelet adhesion. So normally the platelets are inactivated. It will move along with the other formed elements in the blood circulation. But when the platelets get exposed to a disrupted endothelium, which means endothelium is intact all over the circulation, and this intact endothelium is very important in maintaining the blood flow, uniformity, non-clotting, anti-clotting mechanisms like a plasminogen activating factor, etc. So this intact endothelium is very important. So when the platelets will reach an area where there is a disruption in endothelium, the disruption of endothelium will expose a subendothelium. In the subendothelium, there are many collagen fibers present, and in the cell membrane of the platelets, they have proteins. They have receptors for these collagen. So the platelets will get attracted to the collagen fibers, and they will adhere on the collagen fibers. There is another protein which is called the von Willebrand factor, that is also an adhesive protein that also helps in adhesion of platelets to the collagen fibers. And this will give rise to platelet activation. So first, platelet adhesion to the subendothelial collagen, and then this leads to platelet activation. Platelet adhesion actually leads to platelet activation, and when platelets get activated, they develop philopodia. The other nearby platelets also will develop philopodia, and they will be attracted to the site of disruption of endothelium. Then the canalicular systems of the platelets will get dilated. We have already discussed that. And the contents of the alpha granules and dense granules will be expelled into the nearby sites from the cytoplasm into that disrupted area of endothelium. So we have actually studied the contents of alpha granules and dense granules. There is more von Willebrand factor which helps in more adhesion. Then there is more platelet activating factor that will help in the activation of other platelets which are flowing in the circulation nearby. Then there is platelet derived growth factor that helps in the healing process of the disrupted endothelium. Then there is serotonin that helps in vasoconstriction of that particular place so that the blood loss will be minimized. So we have studied all those things. These contents will be extruded in the exterior of the cell of the extruded into the outside of the platelet cell, and these contents will start exerting its effects. We have discussed that the activated platelets, they get adhered to the subendothelium, collagen present in the subendothelium, and the activated platelets also activate other platelets in the nearby areas of the circulation, and these activated platelets will club together, that is called platelet aggregation, and this clubbing is with the help of a protein called fibrinogen. Fibrinogen protein will actually bridge between glycoprotein receptor in the cell surface of the platelets, and this bridge gives rise to clubbing or aggregation of the activated platelets at the site of disruption of endothelium. So platelets adhere to the subendothelium, then more platelets get aggregated together at the site of disruption of endothelium. Then these aggregated platelets will clump together, and that is by the action of certain agglutinins. And this process is called agglutination of platelets. And together this adhesion, aggregation and agglutination will help in the formation of temporary hemostatic plug. So immediately after an injury, the platelets get activated. The platelet will by seal the injury by forming an adhesion between the subendothelium and the platelets. Then the platelets will aggregate together. They will agglutinate together, forming a temporary hemostatic plug. So this temporary hemostatic plug will arrest the bleeding. This is an immediate response to an injury in the blood vessel. Why is it called temporary hemostatic plug? Because it could be washed away easily, not like the original clot. So before clotting takes place, first immediately there will be formation of the temporary hemostatic plug. You can see the exposed subendothelium. You can see the von Willebrand factor, the adhesive protein. Then the collagen fibers, the platelet get adheres to the von Willebrand factor to the subendothelium. Then you can see the fibrinogen bridging between two platelets. So you can see the platelet adhesion and aggregation in this figure. Hope you understood. By now, you should be already able to write some notes on the functions of platelets because we have discussed all. So the first function, first and most important function is its role in prevention of blood loss by helping in hemostasis. We have studied what is primary hemostasis. That's an immediate response after the formation of platelet plug. So it helps in primary hemostasis, then it helps in secondary hemostasis, then platelet help in clot retraction because it helps in production of protein thrombocytopenia. Then role in repair of blood vessels because the platelet extrudes platelet derived growth factor that will help in repair of blood vessels. Then role in defense mechanism because the subendothelium is immediately plugged. The disruption is plugged by the aggregating platelets. It will prevent the movement of dust particles or some foreign materials through the disruption of the blood vessel into the inside of the circulation. So it has a role in defense mechanism also. Next is about the platelet disorders. Bleeding disorders, normally called hemorrhagic diathesis. That's a new term, right? Hemorrhagic diathesis. Bleeding disorders are called hemorrhagic diathesis, and there can be many reasons for this bleeding disorders, and one reason is related to platelets. Then decrease in platelet count is called thrombocytopenia. It will be below 1,50,000 per microliters of blood. And what should be the causes? Decrease in platelet count. Either the production of the platelet should be less or there should be more destruction of the platelets. So impaired production of platelets can happen in bone marrow failure, for example in case of leukemia, or as a result of any drugs like anti-cancerous drugs, then thiazide diuretics, etc. Increased destruction. Example is idiopathic thrombocytopenic purpura. So it can be acute and chronic. This idiopathic thrombocytopenic purpura results is due to the result of immunologic destruction of platelets after viral infection. We have already studied that during viral infection, certain antibodies will be produced inside our body because of the viral antigen. Actually, when virus attacks our cells, there's a viral antigen, and in response to the viral antigen, some antibodies will be produced in our body. And this antibody will not only destruct the virus, but it will also affect the cells. One among them is platelets. So after a viral infection, there is more possibility for the destruction of what? Platelets. That is why dengue hemorrhagic fever. After dengue fever, you will get hemorrhagic fever because more destruction of platelets will occur. So like such things will result in thrombocytopenia, and an example is idiopathic thrombocytopenic purpura. Then increase in platelet count is called thrombocytosis. Then persistent and abnormal increase in thrombocytes is called thrombocythemia. Then inherited hemorrhagic disorder by either structural or functional abnormality of platelets. That is called thrombasthenia. So you should be familiar with all the terms. Okay. That's all about platelets. You should study, write notes regarding the subject. In the next class, we will study about blood clotting. Very interesting topic. So, bye.